it's looking for a specific type of bond. The last type is stereospecific. We've actually seen an example of this when we talked about carbohydrates. We had the alpha amylase and the beta amylase. We said that humans could break down starch and glycogen because that has the alpha glycosidic bonds. The alpha amylase will break down any alpha glycosidic bond. The beta will only work on the beta. So it's not just looking for a specific type of bond, it's looking for a certain three-dimensional conformation. And so those are the different types of things that an enzyme might be looking for. We can also classify enzymes based on the type of job that they do. There are overall 6 major groups of enzymes. A bunch of these, when you look at the word, should make sense. For instance, if you have an oxidoreductase, what type of reaction do you think it might catalyze? An oxidation reduction reaction. You may or may not have noticed a lot of the enzymes that we're starting to talk about end in ASE. That's a good hint that whatever it is that you're talking about is an enzyme. Not a hard and fast rule. So just like with carbohydrates, we said if something ended in OSE, it was probably a carbohydrate. Here, if something ends in ASE, it's probably an enzyme. So we have our oxidoreductases. A transferase will transfer a group from one molecule to the next. A hydrolase is a hydrolysis reaction. You all realize that hydrolysis is when you break a molecule apart, add water to it. You know that's going to be on this test, so I'm going to keep putting that on every single exam until you all get it right. That's one of the words that biologists and everybody uses all the time. It's a good word to know. Isomerases, so whether you completely get it or not, hopefully all of you know that we have isomers of molecules. An isomerase is an enzyme that converts one isomer into the other isomer's form. So maybe it converts the L-amino acid into a D-amino acid. The ligases, I've incorporated the word ligate into my vocabulary, apparently not everybody else has. If you've learned anything about DNA and you've heard about a DNA ligase, it's an enzyme that connects small pieces of DNA together. And so any ligase, that's its job, is to connect small pieces together. Liases, I haven't been able to think of anything that can help you remember what those do. But essentially all of the addition reactions that we had back in chapter 11, where you took a double bond and you broke the double bond and added 2 pieces, a liase would catalyze that. It's something that helps add groups to a double bond, and the double bond gets removed in the process. So those are the basic 6 categories. You should know the categories. You should also be able to take an enzyme if I tell you what it does and put it into one of those 6 groups. So if I say that you have a phosphatase enzyme and its function is to transfer a phosphate group from ATP onto glucose, which type of enzyme would that fall into? Transferase. It would be a transferase. So,
but it couldn't be an isomerase because it's making one thing different?
well, all of these enzymes are catalyzing a reaction. All of them will make something different. It would only be an isomerase. So let's say that I said that you had a, I'm not going to think of an isomerase off the top of my head. A threonine isomerase, and its job was to convert L-threonine into D-threonine. The only thing that it has changed was it changed it from the L to the D. That's an isomerase. Okay, so that's all well and fine. Those are ways to classify the enzymes. The next chunk of material is going to be to talk about how enzymes function. And so before we can talk about how they function, we're going to need a little more vocabulary. So if we look at this, in this picture, the blue is the apo enzyme. The apo enzyme really is just the enzyme. They call it apo because they're talking about the enzyme with nothing else bound to it. We've seen in chapter 18 that sometimes proteins need other things to help it achieve its function. When we talked about the hemoglobin, wasn't an enzyme, but it's the same concept. We talked about hemoglobin needing that heme group and the iron to be able to fully bind to the oxygen. In terms of enzyme chemistry, anything that's required to help an enzyme do its function is going to be either a cofactor or a coenzyme. So both of these things do the same thing. What separates them is what they are. If you have a metal, such as the iron in the hemoglobin example, then the helper is called a cofactor. If the helper is made of organic atoms, your carbon, your hydrogen, oxygen, nitrogen, then the helper is called a coenzyme. So they're both helpers. They both do the same thing. If it's a metal, it's a cofactor. If it's made of organic material, then it's a coenzyme. Another change, up to this point, we've looked at reactions. And anytime we had a reaction, we always talked about the substrate. Or not the substrate, we've talked about the reactants. In enzyme terminology, we're not going to talk about reactants anymore. Now we have substrates. Essentially, the substrate is the reactant that the enzyme is working on. So in the same way that we would have said the substrate, in the same way we would have talked about a reactant, now we're going to talk about a substrate. We've also introduced the concept of an active site. If you remember, that was our Pac-Man. The active site is where things are happening. It's where things are binding and interacting.